Yes, Matthieu, we can.
Okay, just a second. So you can start the recording when you want.
Holly?
It started.
All right, thank you very much. So thank you very much, Colette and Separations, our colleague from Separations, for hosting this webinar today. Thank you for the nice introduction, and thank you all for attending this webinar. So today I will talk about TakaraBio expertise in dealing with challenging samples for RNA-seq. And just to give you a very brief overview of who is TakaraBio. So we have three brands under TakaraBio. Takara, which is a leading brand for many enzymes, including reverse transcriptases and Taq polymerase. So there's also the CloneTech brand, which is indeed the NGS one. So for RNA-seq, but also for DNA-seq, and as you see, also another one which is called Cellatis, which is specializing in stem cell research. So all these brands are under the umbrella of TakaraBio, and what interests us today, of course, is NGS. So what is the very summarized history of TakaraBio and genomics? So it all started 20 years ago, actually a bit more than 20 years ago, with the SMART technology developed by CloneTech in the US. We will see that, of course, a bit later. And this led to the release of the first single-cell RNA-seq kit on the market that was based on this SMART technology. So you may have heard about the SMART ultra low input kit, SMART-seq kit. So it was in 2011. So we continued to develop our NGS expertise in DNA-seq a bit later in 2017, and last year and 2020 we further pushed further our product line for very sensitive RNA-seq with the release of let's say the most sensitive version of our single cell kit, which is called the SMART-seq single cell kit last year, and this year a complete end-to-end solution for SMART-seq from the sample to the sequence. It's very short overview of TakaraBio and genomics, but visualizing our portfolio for NGS, let's say that we have three main technologies: the SMART technologies for RNA-seq with various applications that can be mRNA-seq, total RNA-seq, or more targeted applications, and two technologies for DNA-seq, one which is called throughplex technology, let's say for more standard DNA library preparation, and one is called picoplex technology for single-cell DNA-seq. So today, I will focus only, for the sake of time, on the SMART technology, and mainly on those two applications here that you see, so total RNA-seq and small RNA-seq for challenging clinical samples. And I wanted to mention as well that some of these chemistries also optimized on instruments for high throughput NGS that we have. So as I said at the beginning, TakaraBio core expertise as far as NGS is concerned, in particular RNA-seq, is really about what we call challenging samples. So challenging can be very low input amounts, so down to the single cell, for example, but it can be also degraded samples. So those FFP samples, so from archived solid biopsies, so where RNA is very degraded. And to cope with this kind of samples, so the SMART technology is really core technology of our RNA-seq kit. It's been developed, of course, and optimized along the years for highest sensitivity and reproducibility. And this is actually a very good chemistry for dealing with these complex integrated samples. So the SMART technology, that's an acronym for switching mechanism at the 5' end of the RNA template. When we look at how it works, it's maybe easier than the acronym itself. That's actually a chemistry that allows the full-length generation of cDNA, starting from full-length RNA templates or degraded RNA templates. So we have optimized the chemistry to work with oligo-DT priming when the integrity of RNA is good, or with random priming when the RNA is degraded. So how does this work? So no matter what, oligo-DT or random priming, there would be the first strand cDNA synthesis, and the reverse transcriptase that we use in our kit have this particularity of being able to do template switching. Okay, so when the enzyme reaches the extremity of the template, it will add these extra nucleotides that are not part of the actual RNA molecule, and in turn, these nucleotides would be used by the 5' oligo in the reaction, and for the subsequent second strand generation, just by a simple PCR. The library, the full-length library, double-strand cDNA will be synthesized. So that's a very powerful technology to ensure that all the RNA template is reverse transcribed and, of course, amplified, just because this mechanism occurs only when the reverse transcriptase sees the 5' extremity of the RNA template. So it's been used in our single-cell RNA kits, which, as Colette at the beginning said, are market-leading technologies for single-cell RNA-seq, but also in RNA-seq kits that are really designed to deal with complicated degraded samples, and that's what we will be discussing today. So today I chose to tell you about three main topics. So the first one will be how to deal with degraded samples. I will take two examples, FFPE, so fixed samples, and LCM, laser capture microdissection samples. In the second part, I will talk about liquid biopsies, so biofluids, and how we can analyze the circulating RNA molecules in these samples, and how it can be useful for biomarker discovery. And finally, I will touch a topic which is very important, of course, nowadays and during this pandemic. So this is about virology and how the smart technology can really be very useful for analyzing infectious samples, and talk about viromics, and then I will wrap up. So starting with the first section of this presentation, so it's about FFPE samples and laser capture microdissection samples. So usually these kind of samples are very degraded. So this is let's say the gold standard for researchers that study, let's say, specimens from patients to take a solid biopsy and do these blocks of fixed tissue. This is required in many diagnosis studies, and this procedure actually involve heavy fixation of the tissue, and this is on the right. Those are examples of RNA QC profiles from this kind of tissue, heavily fixed tissues. And just to get you through these plots, you would expect from a high quality RNA to see two very sharp peaks from the 18 and 28S ribosomal RNA. But this is not the case in these heavily degraded samples. Instead, you see a lot of small fragments, small RNA fragments that can be very, very, very small in some instances. So these numbers here that you see, DV200, and those percentages are actually the percentage of RNA molecules which are longer than 200 base pair. And as you can see in this dramatic example at the bottom, 28%, that case of RNA molecules are longer than 200 base pair, which means that the RNA sample is fully degraded. So on top of that degradation, FFPE also induces some chemical modification on the transcript, which is a further challenge when dealing with this kind of RNA. And obviously, due to heavy degradation, the standard oligo-DT priming of the reverse transcription is really not compatible, just because most of the time the poly-A tail is just lacking from these transcripts. So one way of overcoming this is of course to go for total RNA sequencing from these samples and using a random priming. Of course, to get rid of the priming of the oligo of the poly-A tail. So the benefit of performing total RNA-seq, there are many. I just put two of them here. Of course, by using random priming, this is the possibility of getting rid of the poly-A tail, which is not present in this transcript, heavily degraded transcript. But also that's the possibility of capturing all the transcripts which are present in the sample. So potentially non-coding RNAs, so RNAs which are by definition not polyadenylated. So the idea is to get the whole picture of the transcriptome of the sample. And of course, as I said, that's the only way of dealing with poor quality RNA. But in turn, there are besides the challenging nature of the RNA, the challenges also of total RNA-seq lie in the abundance of ribosomal RNA. Because random priming will prime the ribosomal RNA, and ribosomal RNA accounts for the maybe 90% in general of the whole transcript content of a sample. And so for this, there is the need, of course, of having a very efficient way of removing these ribosomal RNA, just to get the useful transcripts in the end. So our solution to that is this kit with a very long name. Sorry for that. So I will refer in the coming slides to this shorter name, which is PICO-V2. So as you see from the name, SMARTer includes the SMART technology that we've seen in the previous slide, and stranded, that states that the strand information of the library is retained. So some specs of this PICO-V2 kit. So that's a very fast library preparation protocol for Illumina sequencing. So from your sample to a sequencing ready library, it's around 6 hours. So this is very fast, and this is a very sensitive product as well, since you just need from 250 picogram to 10 nanogram of total RNA. As I said, this kit uses random priming for the reverse transcription, and as such, it is compatible with RNA from very bad quality. It works also with very good quality RNA, but in our case for today, it's working very well with FFP RNA. So it retains strand information, and of course, it allows you to capture coding and non-coding RNAs. Okay, and included in the workflow, so as I said, that's a complete workflow. Included in the workflow is the removal of ribosomal RNA. We will see how that works in the next slide. So this is the general workflow of the kit. So I already explained the specs on that you see on the right. So I will try to guide you quickly through the workflow of the kit. So starting from total RNA, you see on the left that you perform a typical SMART reaction, but this time using random primers. So what it will do, it will create on the right, here in the middle, a library of double-stranded cDNA that will include also cDNA coming from ribosomal RNA. And the reason why is that the kit is so sensitive in terms of inputment that ribosomal RNA depletion is not technically possible before starting the protocol. So we chose this way of generating the library. So first generating cDNA from all transcript of the sample, and then performing the ribosomal depletion at this stage with a proprietary, let's say, technique, combination of regions that will selectively degrade these cDNA molecule originating from ribosomal RNA. So once this is done just by a final PCR reaction, the useful transcripts actually will be further amplified, and by PCR suppression, the ribosomal cDNA will get depleted from the tube, and you are ready to sequence because the final library is already indexed for Illumina sequencing. It's very elegant protocol, and this is also the reason why it is very fast. So how does the kit performs with these very degraded kind of samples? So this is the picture I showed you before. It is actually the QC of some FFP RNA that we use to benchmark our kit. So you see various level of RNA degradation can be very, very degraded. And on the right, this is the corresponding library profile that we obtain with the PicoV2 kit from each respective RNA samples. As you can see, we get a very nice library, even from very, very, very degraded samples. So all this data is, of course, summarized in a tech note that we have, and this really speaks for the fact that we developed this kit with that in mind, how to give you some very good tools to deal with this kind of samples, very degraded samples. Now, in terms of data that you can retrieve from these samples, so we tested. So this is a busy slide. I'm sorry, but maybe just focus on the box I highlighted here. So of course we analyzed these different FFP cancer biopsies, and those are the number of transcripts that we were able to detect from these samples. So it's above 20,000 transcripts. So this is a huge amount of information to be retrieved from these pathological specimens. And here the colored boxes are actually the Pearson and Spearman correlations across various imputaments. As you see, this is a very robust and reproducible chemistry, even with these highly degraded samples. This is maybe more visual way of seeing reproducibility for these FFP samples. So again, we tested various degradation levels. So 68% of DV200. And have heavily degraded samples, 30%. And as you can see, across various input amounts, so 10 nanograms to 72 or 10 to 90 nanograms of FFP RNA, you see almost a perfect correlation with the Pesant coefficient of 0.98 to 99, which is really great. So in summary, this PicoV2 kit, we have developed it with that in mind, to give you a very sensitive, robust, and reproducible tool to be able to cope with this kind of sample. Just have to mention that respect to the specs I mentioned, so down to 250 picogram of input material, when working with FFP samples, we recommend to slightly increase the minimum input amount to 5 nanogram. We know that it works with lower input amount than 5 nanograms, but this is our recommendation. And the beauty also of the protocol is very fast, very streamlined, and you don't need to do any customization of the protocol for this particular kind of samples. Just follow the protocol, and that should work as nicely as our take note. I want to show you now a real life example of our kit being used for this kind of study. So that's quite a challenging RNA-seq study that use our kit. As you see FFP samples coming from laser capture microdissection. So this is a two-level way of complicating the whole story, starting from fixed material and further inducing degradation just by the technique itself. And so this is the workflow of the work done in this paper by Amin et al. So they process various FFP slides. They perform laser capture microdissection, of course, check the RNA, and went for various assays, RT-qPCR and NGS using PicoV2. And this is the picture of how degraded was the RNA they retrieved from their capture of FFP slides. You see the RIN numbers, so that's an indicator of the quality of the RNA is very, very low. It's slightly above 2, which speaks for very degraded samples. But nonetheless, when they perform the library prep with the PicoV2 and produce those mapping statistics, just as we saw in the previous slide I show you, they obtain what I can qualify as textbook mapping stats. This is actually precisely what we see in our take note. So this is really great. And they also analyze the gene body coverage that they obtain, so to see if they were able to retrieve information, so reads all across the length of the transcripts. And as you see, they were able to have a uniform full-length gene body coverage, so from 5' extremity to the 3' extremity of the transcript. So I think those data are really nice to see, and really strengthen what I told you before regarding PicoV2 and how this chemistry is able really to give you some very good data from very complicated degraded RNA. So I want to switch now to the second part of the presentation. It's about liquid biopsies and yeah, biomarker discovery. So I split that part in two, and we'll start with first with total RNA-seq from liquid biopsies. So liquid biopsies are now kind of trendy way of getting samples from patients. So respect to what I told you before. So tumor biopsies, solid biopsies, FFPE biopsies. This is a very simple way of getting patient samples. So, for example, simply by drawing blood. This is highly non-invasive. This is much better, of course, for the patient. Respect to standard biopsy, it is very quick, of course. It is inexpensive. And very interestingly, also in a clinical context, it allows for multiple sampling, potentially trying to get insight into the diagnosis of a given disease, monitoring disease progression, and eventually response to therapy. So it really opens the door to much more potentially that the standard way of getting solid biopsies. So what we can find in liquid biopsies in terms of nucleic acids. So those are those circulating, so-called circulating nucleic acids. So as you can see, the bulk of circulating nucleic acids is represented by DNA, more than 50%. Of course, you can find proteins, and RNA is really the smallest content of a liquid biopsy, something like 15%. And so in that case, RNA can be found in various compartments of a liquid biopsy. It can be found in circulating cells, of course. It can be found in vesicles, but also you can find extracellular RNA, which is also called cell-free RNA. So simply RNA which is circulating in a biofluid. And in biofluids, you can find various RNA biotypes. I would say almost all RNA biotypes. So for example, polyadenylated transcripts, so messenger RNA, some long non-coding RNAs. You can find non-polyadenylated transcripts like ribosomal RNA, long non-coding RNAs, circular RNAs, small RNAs, microRNAs, a lot of things. And one feature of which is very consistent for each kind of biotype actually is that the input amount can be very low. This is one thing, but also, and was quite unexpected since quite recently, is that the majority of extracellular cell-free RNA is also very fragmented, so very, very, very short. So it's, liquid biopsies, and when looking at RNA, this is also a kind of challenging clinical samples that need a very robust chemistry to perform RNA-seq. And this is an example, very recent example of a study that I used the PICO-V2 kit to perform RNA-seq, total RNA-seq from liquid biopsies. So it is a paper published in 2019. And in that paper, the authors really did quite an extensive benchmarking of the PICO-V2 for that very specific application, total RNA-seq from liquid biopsies. Again, this is a very busy slide, but just to show you that they analyzed various kind of liquid biopsies, so plasma samples, vesicles, purified vesicles. They also analyzed urine, which is also a biofluid, and from all these kind of biofluids, they perform RNA-seq with the PICO-V2. this is an example of reproducibility they were able to get from two different biofluid samples. So EPRP refers to platelet-rich plasma. Okay, so it's plasma rich in platelets, and PFP is platelet-free plasma, so plasma depleted from platelets. And in both cases, they were able to show highly reproducible libraries generated with PKP2, which is, of course, great because we are talking about patient samples. As you see here in that slide, they were also able to detect various kinds of RNA biotypes. So those are just some of them. Long non-coding RNAs, RNAs which have no real function, might be interesting for biomarker discovery, pseudogenes as well. But the message of these graphs, as you see, so each column, so to say, is a different kind of biofluid, of liquid biopsy. And you see that, you know, the content of RNA is different for a given biofluid. They were really able to uncover that biofluids are heterogeneous in terms of transcripts they contain, and that really raised the question, or at least the possibility of let's say using this information, these various biotypes discovered in various biofluids for biomarker discovery in various disease states, for example. And I quoted the final sentences of the discussion, and this is the conclusion they came out with. Indeed, biofluids and RNA-seq, total RNA-seq from biofluids is really a promising way of diagnosing diseases and monitoring disease development and therapy. And one bottleneck of biofluids is, as I said, the low input amount of material, and of course the low input amount of useful information you can retrieve from these very small samples. And this is something that they were able to overcome by using PICO-V2 for total RNA-seq from these samples. So this is very exciting. So another topic related to liquid biopsies are small RNA-seq. So small RNAs, maybe microRNAs in particular, have been regarded for quite a while now as biomarkers, and this is something that we can tackle as well with our portfolio. So I put the workflow of a kit called Smarter Small RNA-seq kit that we offer for small RNA-seq. Straight away you can recognize the smart reaction. There are small tweaks, of course, respect to the standard smart reaction. But anyway, that's a kit which is specifically designed to capture small RNAs, so below 150 base pair, so down to 15 base pair, starting from total RNA. This is a very sensitive kit. It can go as low as 1 nanogram of total RNA, which is very low when we are talking about small RNA sequencing. And one particularity of this workflow is that it's a gel-free protocol. So most of these small RNA-seq strategies beside ours include at some point a gel based enrichment of small RNA transcripts at some point. So it's quite time consuming, can be tedious, and of course when you have to do this manually, you may or may not select the very precise band of interest. So that's challenging. In our case, all happens without having to select small RNA, and the site selection is actually done only by magnetic bit purification. So it is a way of highly facilitating your small RNA-seq studies. So this is a kit we also developed with that in mind. So being able to work with these kind of samples, as you see, plasma samples, serum, and to allow you to be able to discover some biomarkers in these biofluids. So to make it very short, we looked at microRNA expression in these kind of samples. We were able to detect them, and very importantly, again, when dealing with patient samples and biomarker discovery, in particular, reproducibility is key. So this is what we visualize here in these dot plots with almost perfect correlation among replicates. So this is a highly reproducible chemistry for small RNA-seq. Now some examples of how the small RNA-seq kit has been used in that context. So I think this is a very important work, very recent work published in Cell last year, where the authors actually did an extracellular RNA atlas from biofluids, and in that case they really focused on small RNAs. As you can see the numbers, this is a massive effort to contribute, to document those liquid biopsies and to document the small RNA species which are present in these samples. And yeah, this is freely available to the scientific community. So a massive work using small RNA sequencing for liquid biopsies. And this one is very recent. So as you can see, this is a bioRxiv paper. So take it with a grain of salt. But I think the message is very interesting. So as you see from the date of the bioRxiv paper, it's been published end of this April, so it's very recent. But here the idea is that the authors made small RNA sequencing from various cancer specimens. You see the number of cancer patients. They looked at extracellular vesicles, so liquid biopsies, and again, to summarize the whole study, they were able, by looking at extracellular RNAs, to use some of them as early biomarkers of cancers from liquid biopsies. So again, this is very exciting. But what is even more exciting is that they really focused on non-annotated small RNA transcripts. So not what is already known, but what is usually discarded in the RNA-seq study. So this is again a very exciting story in that topic. Finally, the third part of the presentation is about let's say virology, so what we can call viromics, and how RNA-seq can be done in that context. I of course chose to to talk about COVID-19, so the SARS-CoV-2 virus. So this is a pioneering study made in China, published in March 2020, where the authors actually sequenced the full-length genome of this new virus with the PICO-V2 kit. So it was quite exciting because it was the first study to provide full-length sequence information of that virus, and at the time, of course, it was done by a metagenomic approach, without any prior knowledge of what could be present in the samples of these patients. So from this study that characterized the virus, things have evolved, and now we are at the stage where massive detection of swabs is performed, and most of the times there is some RNA left over from this type of assay, and this leftover can be used for other kind of assays, and this is, for example, NGS RNA-seq. And the idea behind that is to perform RNA-seq to understand the viral genome diversities, to get full-length sequences of the genome, to pick some mutations potentially happening, and this information is in turn used to track infection path and to inform public health authorities. So this is something called genomic epidemiology, and this is something which is very important for public health authorities. In that case, the main challenges are the very low amount of the RNA left over from the swabs. RNA, I said, is quite often degraded. RNA is very degrading all the time, and of course, because the idea of the NGS assay is to get SNP detection, the chemistry needs to be very robust and very sensitive to give this information. This is the picture of how PICO-V2 has been used for viral genomics. You see that many different viruses in various ways have been analyzed by this kit. I've put in blue two examples. So the one I just told you about, SARS-CoV-2, which has been done on leftover of COVID-19 testing samples, and this study is actually a genomic epidemiology study. It was done by target enrichment protocol, so putting down the virus sequences from the PICO-V2 library, and also this one focusing on HIV. So why I chose to highlight those two studies, let's say, so because they are very recent and they're from the same group, and I think that the take-home message of those two studies are very interesting. Starting with the first one on HIV, you see this is very recent, so it's just been published. So here, this is a massive study on more than 1,600 plasma samples of HIV patients. You can see on the graph the full-length coverage of the HIV genome, which has been possible to get with PICO-V2, and the various colors are actually the viral load of the samples, so the number of copies, HIV copies per ml. And here, the authors were really able to show that they could get full-length HIV genome coverage from low viral loads, so 1,000 copies per ml is quite low, and this is actually the standard for genotyping based test currently approved by FDA. But on top of that, what is very interesting in this study is that they were able to cut down the cost of the assay down to £30. So I don't know what it is in your currency, but this is somehow quite inexpensive, and this is in the standard of existing genotyping tests. Again, on top of that, sequencing versus genotyping does not offer the same wealth of information. And in their case, beside being able to say, yes, you are infected by HIV, they were also able to get some specific mutations of the HIV genome, which were known to be responsible, for example, for anti-retroviral drug resistance, which is key to inform, again, public health authorities. And the very last study, it's very recent, June the 2nd of this year. So same group worked on SARS-CoV-2 sequencing. So far, they sequenced more than 400 patients. And what they were able to detect is some, let's say, variability in the genome SARS-CoV-2, which was more than what we expected until recently. And they were also able to give insight into the, let's say, the epidemiology of the infection by uncovering wide transmission bottlenecks. So that means that each single patient contained a lot of SARS-CoV-2 genome variation, and this is also very insightful. So to summarize, clinically relevant samples like FFPs or solid liquid biopsies, pose many challenges to researchers. Most of the time, all these kind of samples are degraded and present in in minute amounts, which means that the usable material is very, very low. So to cope with that, of course, sensitivity, reproducibility, robustness of an RNA-seq protocol is needed to be able to provide useful data from these patient samples. And I showed you today in various examples, FFP, liquid biopsies, and viral samples, that the SMART technology was a very sensitive and robust one to be able to process these very precious samples and potentially make breakthroughs. If you want to learn more NGS about NGS solutions from TakaraBio, you can. It was a very small insight into our NGS offering, and that's good science.
Thank you very much, Matthieu. I don't know if anybody has specific questions. If you do, you can post them in the chat so that Matthieu can maybe answer them for you. No questions yet, Matthieu. I think you've been very clear then.
All right. So if you have any question, later, don't hesitate to reach out to Separations, and I'd be also happy to answer them. So thank you very much for attending this webinar. Thanks again to Separations, and wish you a very nice day and stay all safe.
Thank you very much, Matthieu. And maybe if you can just share that HIV publication with us, that would be great for some of the participants that have contacted us and asked us to share that.
Of course, I will. No problem.
Thank you. Thank you, Matthieu.
Thank you very much.